Hello, everyone. My name is Raquel Manzano. I am Spanish, but I come from UK. I am a part-time bioinformatician and PhD student at Cancer Research UK, the University of Cambridge. And today I'm here to talk about my story or my adventure with Nextflow and the development of this pipeline. I want to give a bit of background because I work in cancer research, so I just want to make sure that you understand why I'm actually doing this. So in cancer, mutations are quite important. You can do a lot of type of analysis, and they can be more or less sophisticated. So it's very important to identify drivers, so that can give us an idea of maybe what kind of drug treatment can be good for the patient, or what sort of things we can use, and then the mutational signature analysis that tells you the biological processes that might be driving the tumor growth. And then you can be very sophisticated and go for a subclonal deconvolution, and you can do this even with bulk sequencing data. And that means that you can actually go into your sample of the tumor and try to deconvolute what kind of different cancer cell populations you have. And just another thing that I want to say, so it's clear, in cancer we talk about germline and somatic mutations. Germlines are the mutations that you are born with, and then the somatic ones are the ones that you acquire later, and these are the ones that you want to study in cancer. So how do you do this? So it's fairly straightforward now to do this kind of analysis. We have JTK, the Genome Analysis Toolkit, which is great. It provides best practices to do so. And of course, we have SAREC that you should use because if you're here for variant calling. And actually, a great part of my pipeline is based on that. But for RNA, because I wanted to do more, right? Like everyone is doing variant calling in DNA. Now I want to take it a step forward. That is what you do when you're doing a PhD, try to do something else. And I said, okay, I want to go mutations in RNA sequencing because we have a lot of data. We have very big data sets that have DNA and RNA. And I thought, well, it would be cool if we can combine that information and we can know and we can do some sort of integrated analysis, even doing the subclonal deconvolution that I was talking before, and see if you can know a bit more about that intratumoral heterogeneity, if those mutations are being expressed or not, how does that work out. But RNA is more challenging. And it's challenging for technical reasons and for biological reasons. So we have an equally distributed coverage. Of course, we have upregulation than regulation. We have higher levels of duplication. And then we have very cool biological things, that is alternative splicing, polyadenylation, and RNA editing, which happens normally in our cells. So it's something normal that we can find. Well, that means for the purpose of a bioinformatician that you will find a lot of artifacts. If, for example, you do a DNA analysis and you end up with 100 mutations, in RNA you might find yourself with 1,000 mutations looking at the same. So it's like, okay, so now what do I do with these 10x more mutations that I find in RNA? Are they worth it? Are these artifacts? So while I was working on this, then this paper came across. So this paper is from Gadi Getz's group in the Broad Institute. And they developed this RNA mutex pipeline that was very simple, but quite straightforward, where you can do your variant calling with RNA using the match normal. They were using mutex, so mutex 1, not mutex 2. And then they were doing some processing for the RNA, where they were doing some filtering. And this filtering included all these steps. They did a realignment. They would check on the known RNA editing events because there are databases out there. They would annotate them so you can remove them, so you end up with a nice and clean set of mutations. Some other filters. If you are interested in any of these, just let me know, and I can go deep into it offline. So my goal for my PhD was actually to, what I said before, right? I wanted to use DNA and RNA to call somatic mutations. In my case, I work in a breast cancer research group and look at intratumoral heterogeneity patterns. So my roadmap was straightforward. So I would reproduce what RNA mutex did. I'm going to update the pipeline a little bit because I felt that using Mutec 1 in this moment was, there is Mutec 2 now, and there are tools that do more. So I wanted to update that and tune a bit more the parameters. And another thing was like RNA Mutec was a bit scattered. It was like MATLAB script, Bash script. So for me as a bioinformatician, it was a bit difficult to make it work, and I felt, well, if I'm going to be using this, I may as well use my time and do something more reproducible. And then use this pipeline to analyze the cancer samples that I wanted to. Easy, that is what I thought. Done, I will do it. So I did develop a Python pipeline with this, and I did nothing as close as Nextflow, but I did some, it was working very well with my HPC system, and I could resume as well and do some stuff, but again, nothing as fancy as Nextflow. I didn't know yet what Nextflow was, and then I updated the workflow, I did the analysis, everything was working, and I said, oh, I did this maybe too fast. So then, right, I arrived to the stage that now I have to do the analysis of all these samples, and yeah, I need to make it scalable and reproducible, right? And I was thinking, yeah, I have my system. I know it works in my setting, but then, you know, I felt this itch where, but if I want to publish this, are people going to be able to use what I have done? And yeah, I thought, probably not. So I started trying to search what I can do about that. And that is when, okay, I started, of course, Googling scalable and reproducible research, bioinformatics, blah, blah, blah. And that is when I went down the rabbit hole, and I found Nextflow. I think the first hit was the RNA bar pipeline that already follows JTK best practices, which you say, oh, great, I can do this, but I actually want to use Mutec and other variant colors. And that is where I found Sarek, and then I read a bit about Nextflow, and then I clicked on the Slack join, and then, yeah, that was it. I think the first, I wrote in the new pipeline, like, I want to do this, would it be okay? And I think it was Maxim, the first one that answered, and he said, of course, yes, we can do this, it's going to be fine. I said, awesome. So 1 year after, because I thought, oh, I can do this fairly fast, but yeah, 1 year after, I have my TÜV plot as well, my TÜV graph for my pipeline, and now I have developed this DNA and RNA somatic variant calling. It's called DNA and RNA bar. It is in GitHub now. It is still in development, but I'm really hoping for a first release next month. That is what I've been saying for several months. But I trust this is going to be the good one. And part of the pipeline, again, I said, is a lot of Sarek, but I didn't need, because Sarek is very fancy and has a lot of features that I actually, at the moment, I didn't know what to do with them. So I just said, okay, I want just the variant calling superflow, and then for RNA bar, I take the best practices, and I did my little Frankenstein of all these fantastic NF core pipelines. And I did add quite a few things, because I said, well, now that I'm doing this, and it's going to be scalable and reproducible, I want to do something end-to-end. I want to be that person that says, just press that button, and then you get the results. I can go on holiday, and everything is there. So I'm going to talk a little bit about the new sub-workflows. I have restructured things a little bit. I welcome a lot any feedback, criticism, because I've been trying to do things a bit differently, because as a good bioinformatician, I can be opinionated, and I like certain structures, and I have done some extra sub-workflows of the sub-workflows, because I felt the first time I looked at Sarek, I felt, wow, this is a lot of code just in the main one, and I felt a bit lost, and I have done some restructuring of that in my pipeline. And I have included things that I think that are interesting. So for example, when you finish with Sarek, you finish with a bank of VCF files. So I have included a new sub-workflow that is for the normalization of the VCFs. If you are familiar with variant calling, you might find yourself that when you are trying to put everything together for SNVs, so for single nucleotide mutations, it's fairly easy to just compare them. But if you run into indels, it might be a bit more complicated, because they can be annotated differently between the colors. So I have a small sub-workflow to normalize all that, so everything should be in the same coordinate or left aligned, et cetera, et cetera. And then I have also implemented a consensus, so you end up not with a bunch of files that you don't know what to do, but with an actual final file that is in a MAF format. MAF format is specifically for cancer. It was designed by ICGC, the International Cancer Genome Consortium, and it's a mutational annotation format. So it's a table file, but it is standardized, and you can add there all the columns that you want. And then it is very easy to play around with it because it's a TXT file. Then I have also included some math filter in sub-workflow, and I have tried to do this as flexible as possible. In my opinion, I have put the filters that I think everyone should be doing in cancer, like NOMAD, maybe some VAFs. You can tweak that if you want, and again, I very much welcome any input on that. If you think that something should be added, we can talk about it, and I can do it. And then I have also done this realignment step that RNA Mutec used to do. So this is more related to what I was saying before about the restructuring and doing things in a way that can be as reusable as possible, because since I was putting a lot of effort into this, I actually wanted to make it good that you can just take this and take what you like and run it. And everything is similar to SARX. You can start from FASTQ files, BAMs, CRAMs, VCFs, and math files if you want. So you can just do the filtering if you have your math file and check how it looks like. And just in case you're wondering about the X that is there in the middle, it's just because if you have DNA and RNA, and you want to leverage that information, in the consensus step, you can use the DNA and RNA. Well, the way that consensus works in variant calling, sorry I didn't mention that, is that I have my 3 callers, and it's a very simple voting system. So if 2 of the callers are calling that mutation, they say, okay, this is a good mutation. And in the case, if you want to do what I call a rescue step, if you also have the DNA or the RNA, that is sort of orthogonal validation. So I add also that, so it gives an extra vote there. And if it is called in DNA and RNA, it should pass. So what's next? So I did say at the beginning, first release coming very soon with a preprint that is also coming very soon, working very hard on that. At the moment, what I've been working, I was in the hackathon, which was fantastic. Apart from doing my first NF test and doing a PR to SAREC that was successful, I also had time to work on my test profile and things. And it was great because at the end of the hackathon, I did get the message that the pipeline completed successfully with the test profile. So, wow, that was nice. So all this pending of review and pushes, but it will get there. That's what I'm saying. It's coming soon. And now after this, I will finally do what I should have done a year ago, testing the scalability and run my analysis on breast cancer data sets. And then of course, I am a multitasking person. I like to do things on the side all the time. So I did like a lot NFTs. I think it's great. So I would like to implement this in my pipeline as well, maybe for a second release. And also the sub-workflow recycling. So I want to, I've been talking with Maxime and some other people about maybe you can put this in SAREC as well. Or for whoever thinks it's interesting, again, come and talk to me. And then I am also very eager to spread the Nextflow in Cambridge. So I'm part of the bioinformatics training unit. And I'm trying to prepare it. You don't always have time to do all the things that you want, but I'm really hoping that I can get this done fairly soon. And finally, just a very big thank you. This is my lab there in Cambridge, to my supervisors, Carlos and Oscar, and to Gadi Gates, he also gives the input. And then, of course, thanks a lot to Secure Lab. I have Maxime. Maxime has been great. He is my official supervisor because I've been part of, well, supervisor mentor. I've been part of the mentorship program at the Nextflow, well, NF Core, and that was fantastic. If anyone is thinking about doing it, do it. It's really great. And I also want to say a big thank you to the Open Bioinformatics Foundation because they found me to come here, and it was great. And, you know, the biggest thank you to the Slack community because you guys are great. You won my heart, so thank you very much.
Awesome. Thanks again, Raquel. We caught up a little bit on time, so I'm going to choose one question here from Adam. Can you compare multiple RNA samples versus one DNA sample? And are there any plans to add detecting fusions?
That's a great question, Adam. And the answer is, I don't know. I would have to test that, but I think that's actually quite interesting. Yeah, we can talk about that. And there was a second question?
And are there any plans
fusions.
to add detecting fusions? Yeah.
Fusions. Yeah, not at the moment, because we need a first release first, but I think, yeah, that would be great, and that would be a great collaboration with the RNA-seq pipeline as well, because I think that, yeah, RNA-seq with fusions, it would be great to have it as well.
Awesome. Well, thanks again to Raquel. And